divides, how this cell grows, how it's going to be basically preparing or new. So most of the division results in genetically identical daughter cells. This is like mitosis. Okay? In order to replace the cell, it has to be the same cell, exact copy of DNA. Yes, mutations can cause errors, but the role for this cell division is to be like exact copy of DNA. And here in mitosis, in this cell division, the distribution would be of this identical genetic material to the daughter cell, the DNA. So you go from one cell to two cells, identical copy of DNA. Mitosis. The DNA is passed from one original cell to the next with remarkable fidelity. Again, with the exception whenever there are mutations, but that should not be the case. There are some processes for like proofreading and so on. In case there are some errors in the DNA. But the goal is to have this remarkable fidelity, and you have the exact copy of DNA from the parent cell to the daughter cells. So whenever you mention here the genome, that basically is stating that the whole DNA in the cell, that is the genome. The DNA in the cell constitutes this cell's genome. So this genome consists of a single DNA molecule that is common in prokaryote cells, or a number of DNA molecules, those are the chromosomes, in eukaryotic cells. These DNA molecules in the cell are going to be packaged into chromosomes, and eukaryotic chromosomes consist of chromatin, which is a complex of DNA and protein, histones, proteins, coming together in order to wrap DNA around, in order to basically like tightly pack DNA so it can fit inside the cell, inside the nucleus. So here's basically what you have a picture of that. The chromosomes right here, DNA staining. So you have it dyed, so now you can see that, and that's basically destroyed by water and so here's basically the different phases of DNA packing. So you have here the short region of DNA double helix, and then you have like this string, you have the proteins, the histones, and then you're going to form this complex of proteins and histones and DNA, and you have these nucleosomes, and then it's going to extend it like different regions, and then it's going to be condensed the whole DNA section in order to form this chromosome right here. So that would be the chromosome. And here, that is the centromere. That is the waist of the chromosome. That is where they're like more highly associated, tightly packed right there, condensed. Those are the centromere. As you can see here, those are the sister chromatids right there. That will tell you that it has been to the S phase. In other words, the DNA synthesis. So this arm of the chromosome is the exact copy of this arm of the chromosome, ready for cell division, to give one to one cell, to give the other one to the other cell. Okay? If you were only to see one of them, only one arm, then it has told you that it has not undergone the S phase, the DNA synthesis, the exact copy of DNA replication. Okay? Because you see two, you see the sister chromatids, so therefore that are then ready for cell division. You can see this as here we have the loose DNA, uncondensed DNA, right? That is the S phase. In this S phase, that's when you have the G1, the whole interface, the whole G1, the whole S phase, the G2, that's the whole like interface, okay? The S phase, that's where you have the DNA replication. G1, G2, cell grows, and then you have the mitotic phase. That is whenever the DNA is going to divide, whenever the DNA condenses, and that's what you see here, the condensation of DNA, identical copy, one to give one to each daughter cell. The interface and the mitotic phase. Really amazing how it goes from here, all uncondensed, to really like highly tightly packed DNA. So it can be divided. Really amazing, really amazing. If I could give you an analogy for this, think about like all this loose, uncondensed DNA. Think about, for example, like a fishing line that is like 150 miles long. Think about, like, for example, from here to Galveston and back. And then you highly tightly packed, so condensed that it fits a size of a basketball. That's pretty much what is happening right here. This long DNA that is inside your cells that is going to be like so condensed so it can fit inside the nucleus and the chromosomes to be separated. And you do that in the process of like 1 hour. Really amazing what your cells are doing, all this machinery that is like going from here, from your DNA in the interface in the G1, so it can be expressing your genes for your cells to function, whatever they need to do, expressing the proteins and all of that, into going to the mitotic phase to condense it, tightly pack, and put it into the identical copies, and then pass one copy in another copy to the other cell. Really amazing cell division. And you want to think it like even more amazing, think about this whole process. The total length of DNA present in one adult human is calculated by this. The length of the base pairs, 134 micrometers, 3.4 Angstrom. And then the number of base pairs per cell, that is like 6 billions. And then the number of cells, that's here like 10 trillions, but actually I think it's more like 30 billions, that number changes. And then you multiply all of those, and then you have like 20 trillion meters of this DNA. That amount is equivalent of going like nearly 70 trips from the Earth to the Sun and back. That's like almost DNA that is inside the human body, and you have this machinery to process all of that, highly pack, condense it, and then divide it, cell division, to fit all of that inside the chromosomes, inside the nucleus, inside the cells. Really amazing, really amazing. So, every eukaryotic species has characteristic number of chromosomes in each nucleus. You have these different cells. You have the somatic cells. Those are like soma, body, the cells in your body. So all the cells in your body except for the reproductive cells. So these are the non-reproductive cells. The cells in your skin, the cells in your liver, the cells in your lungs, all of them. Somatic cells. They have two sets of chromosomes. And then you have the gametes, which are the reproductive cells, the sperm cells, the egg cells, and they only have one set of chromosomes. So they're halfway. So somatic cells, two sets, and then gametes, only one set. So I have an example of human somatic cell. I mentioned a couple of them. With human gamete, I mentioned all of them, sperm cell, the egg cell. Okay. So in preparation for cell division, the DNA is replicated, that is the S phase, and then the chromosomes condense. That is the whole mitotic phase, right? They're going to be like so condensed, highly packed. So each duplicated chromosome has two sister chromatids, so they're going to be identical, and they're joined identical copies of the original chromosome. So they have to be the same. They are joined by the centromere to the chromosome. So the centromere, that is where the two chromatids are the most closely attached, joined together. That is the waist of the chromosome. So here you have this picture right here. You have the cell, or here inside the nucleus, you have the chromosome arms, and you have the centromere right here. Okay. Then you have the chromosome duplication, which including like DNA replication and condensation. And now you have here this chromosome with the sister chromatids. Then you have separation in here to have the chromosome inside the daughter cells. So how many chromosomes are in the middle cell right here?
2.
How many?
2.
1 or 2?
2.
Or 3?
It's
Or
2.
4?
It's 2.
4, yes, correct. There is no 4
Okay,
right
so
here.
no.
How many chromosomes right here?
1.
1. How many chromosomes right here?
1.
How many chromosomes right here?
1.
How many chromosomes right here?
2.
None? Wait.
1.
Because they're together, baby.
They are together. These are sister chromatids. They are joined together, okay? It's one chromosome, but two sister chromatids. Once the sister chromatids separate, they become chromosomes, okay? Think about it like this. You are a baby once you live inside your parents' house. Once you go outside, you're an adult, okay? Once you leave outside your house, once you're 18, okay? But before that, you're dependent. So they're dependent, okay? They're dependent. They're one chromosome.
That's one chromosome, and then they split into two.
They splits, the sister chromatid splits, the sister chromatid splits, they become the chromosome. That's a good analogy, yes. So one chromosome, two sister chromatids, they're identical, that's the key word, they're identical. Yes?
So the way I think about it is that you're in one family and you have a sister. When you both get married, then you have two families.
Yeah.
So that's just one family as a sister chromatid, right? And then when you split, go into your different families, you have two families.
You make your own family once you get out, once you get married, okay? But But inside here, one family, one chromosome. Once they split, once they get married, once they get family, then two chromosomes, two families. Okay? So do not get confused by that, okay? And you will see these kinds of questions on the exam. In different ways, you will see. So during subdivision, the two sister chromatids of each duplicate chromosome separate and move into two nuclei, right? So now you have like two differences. Once they separate, then the chromatids are called chromosomes. But once they separate, once they are together, it is one chromosome. Okay. So, do that. One chromosome right here, one chromosome, two sister chromatids, and then here they separate, and now they are two chromosomes, two daughter cells. So, what does this tells me, and what does this tells me if both are one chromosome? This one has not gone the S phase, the DNA replication, DNA synthesis. This one has gone over the S phase, the DNA replication. So this tells me that this cell is ready to do the cell division, the mitosis part. Okay. So it has been replicated, DNA synthesis, the S phase, okay, ready to separate. All right, so eukaryotic cell division consists of the mitosis, that is the division of the genetic material in the nucleus, DNA, and cytokinesis, that is the division of the cytoplasm. Gametes that are produced by a variation of cell division, that is meiosis. We'll cover this later on the next module, okay, or the next section. So gametes that are produced by meiosis. But the division that cell division that we're seeing right now, it is mitosis. Mitosis, the separation of the material DNA, the nucleus, and cytokinesis, the separation of the cytoplasm. So if a cell of a rice plant has 12 chromosomes, how many chromosomes will be in the cell after it undergoes two cell division? So it is first 12 chromosomes and then 1, 2. How many?
12.
Sure?
Yeah.
Basic math.
Oh, no.
12, 12, 12.
24.
No. That would be
Remember, it
confusing.
never changes, right? Yes, yes. So the answer will be 12 chromosomes, right? It never changes. Do not get confused by those kinds of questions. In this cell happens mitosis, in this cells happen meiosis. What is the chromosome number? If it is mitosis, equational, always the same. Those number of chromosomes, they will always be the same. So if it has 12 chromosomes, it will have 12 chromosomes regardless of the number of cell divisions. Meiosis, on the other hand, when we see that, it is reductional. Mitosis equational, the same number, meiosis reductional. If it begins with 12 and it happens meiosis, then now it's going to be half. It's going to be 6. Okay. In meiosis, reductional. Both mitosis and this equation, always the same. So if it has 12 chromosomes there, if it has like 20 cell divisions, the number of chromosomes will still be 12. Okay? If there is another organism, it has 6 chromosomes, and it happens mitosis, and it happens 5 cell divisions, it will still be 6 chromosomes, equational. Okay? So whatever you see there, 12 chromosomes, 6 chromosomes, 40 chromosomes, whatever it is, if it is mitosis, it will be the same. 12, 12, regardless of number of cell divisions. Yes?
So, like, if it's like 3 cell divisions and it's 12 chromosomes, and we're talking about meiosis, would it be like 12 divided by 3 or 12?
No, no. For that one, just reductional, just the number of whatever it is, in this case 12, then that will be 6, like 4 meiosis, right? But on that one, you can see here, that cell goes this particular number of cell divisions. On meiosis, once you have the gametes, then they are going to go over there and try to fertilize or be fertilized, right? They're not going to keep dividing it undergo meiosis. So once you produce the gametes, then you're going to use them or utilize them as the end goal. Okay, good question. So the cell cycle consists of the mitotic, that is the M phase, that includes the mitosis and the cytokinesis, right? The division of DNA, division of cytoplasm, and the interphase. That's basically what includes the cell growth and the S phase, the DNA synthesis, DNA replication. That is the coupling of chromosomes in preparation for cell division. So here basically the interface. This is about like 90% of the cell cycle. We divide into these subphases. The G1, that is the first gap, G1 phase. That's actually where gene expression takes place, where the DNA is being basically utilized, transcribing genes and so forth, protein expression. The S phase, that is the synthesis part. Yes, question.
I thought the G1 fusion was when the cell rests.
You said what?
The cell rests.
Rest. No, I think you're talking about the quiescent state. That would be the G0. This one, the G1, is cell growth, cell proliferation, but also for the gene expression, where that DNA is going to be expressed to make those proteins that it needs to utilize for the cell to perform its particular function. Okay. And then the S phase, that's where the DNA replicates. You have the DNA replication, the DNA synthesis. And then the G2 phase, that is the second gap, that's where this cell continues to grow, and making sure that everything will be ready for the cell division. That is, okay, you have this number of mitochondria for this cell, you have this number of mitochondria for this other cell, okay, you are good. Are there any errors on the DNA synthesis part, DNA replication? No, you're good, okay, you're ready to move down into the mitotic phase. Yes, there are some errors here on the DNA replication, okay, let's fix this, or basically induce some cell suicide or like apoptosis, right? It's going to basically kill the cells. So it will not basically propagate these mutations that it happens here on the DNA or DNA synthesis on the S phase. Okay. So this G2 phase is really important for that, checking corrections, checking for errors, correcting those errors, make sure that it's ready to divide, make sure that everything is ready. Once it has been like growing, exact number of organelles, so it can divide. Okay. So the cell grows in all these three phases, but chromosomes are replicated on the S phase. So that's what you have here, the interface. This is G1, cell growth, gene expression. The S phase, DNA synthesis, DNA replication, exact copy of DNA. G2 phase, continue to grow, and then check for errors, make sure that there are no errors right here on the DNA synthesis, and then right back. And then you have the mitotic phase, which is the mitosis, separation of DNA, and the cytokinesis, separation of the cytoplasm. And then you have the two resulting daughter cells. Mitosis. Okay.
They're the what?
The two resulting daughter cells.
Oh.
Identical daughter cells. Exact copy of DNA. Because of this DNA synthesis. Okay. Mitosis. That was the here. Interface, G1S and G2 phase. And then here the mitotic phase. The prophase, prometaphase, metaphase, anaphase, and telophase and cytokinesis. So, mitosis conventionally divided into these 5 different phases, and cytokinesis overlaps the large stages of mitosis, the telophase. Then you have the G2 phase of the interphase. You have here the loose DNA or the uncondensed DNA. You can see here the nucleolus. What is the function of the nucleolus? Remember that? What is the function? Close. Remember that, the function of the nucleolus? The one over that on the second module? Organelles? Ribosomal RNA, right? That is the synthesis of ribosomal RNA, right? Which is complete for the ribosomes, important for making proteins. And here you have the nuclear envelope right there, uncondensed DNA. And then you have here the centrosomes. Remember about this? We went over this in the last couple sessions. That is the central burst. Okay, so you have the centrosomes here, perocentrioles right there, perocentrioles. That is the G2 phase, cell growth. Now you have here the prophase. Now it goes to the mitotic phase. In this prophase, you have here these centrosomes. Now they begin to migrate. Okay, opposite ends. So here you have one perocentrioles, here you have another perocentrioles, and the microtubules begin to grow. Okay. Microtubule organized center, microtubule organized center. Then here you have the condensation of the DNA of the chromosomes. Uncondensed, condensed. See here, you don't see the nucleolus anymore. And here for the condensed DNA, the chromosomes, you see the centromere, the waist of the chromosome. The prometaphase, basically you have the fragmentation of the nuclear envelope. You don't see that anymore here. We have here further condensation of the chromosomes. Here you can see that they're like longer. Now here like they are more defined. So further condensation. Now they're like more highly packed, condensed DNA. So nuclear fragmentation that allows for the microtubules not to invade the nuclear region. They come and they're going to find the kinetochores in the chromosomes. They're going to bind it. Okay. So here you have some chromosomes that are bound, kinetochores, bound to microtubules from both ends, but here you only have one, the other one is not. The other one is not. Right? So in this phase, that's where the microtubules are going to go and invade the nuclear region, bind to the kinetochores of the chromosome. But they're not in the middle yet. So they're going to be placed in the middle once these kinetochores on the chromosomes that are bound to these microtubules on both ends. So there is like equal push and tension from both sides that they're going to be placed in the middle of the cell. So that is the metaphase plate. Once they are placed in the middle of the cell. Why? Because of the tension, push tension between the microtubules and kinetochores that are bound in the chromosome. Then you have the anaphase, and that is the separation of these sister chromatids that are going to be separated, they're going to be migrating into the opposite end of the cell. Anaphase, separation. Then you have the telophase and the cytokinesis. Here, you will have now the centrosomes no longer yielding the microtubules to go and invade. Now they have been like separated, so they don't need that anymore. And you will see the uncondensation of the DNA, begin to uncondense, okay? And you will see the nucleolus formation as well. Once you see here nucleolus formation right there, then that tells you that you are on the end phase of this mitosis in the telophase. Okay. Here, basically, I'm showing you what happens in the metaphase, going to the anaphase. So what you have here is in the prometaphase, you have the chromosome, okay? And you have these centromere, and you have these kinetochores that are bound to the chromosome in the centromere. Okay? And also, joining these sister chromatids, you have the cohesion proteins right here. These cohesion proteins, they are binding together these sister chromatids. Then, you'll have the microtubules that are being sent by the microtubule branching center in order to bind into the kinetochores. So you can see here, they're bound right here on this side, but not on this side. So there is this lack of tension. So it's only bound to one side. Once it is bound to both sides, then there is equal push, equal tension. So now they are placed in the metaphase. Think about like that, like tug of war, right? You're pulling from both ends, they are equally strong, they're going to be placed in the middle. So that is the metaphase. Once they are placed in the middle, once they're like into the anaphase, once all chromosomes that are placed in the middle, then that sends a signal to the separase. That is a protein that will come and bind and cleave the cohesion proteins. It will cut them. Once it cuts the cohesion proteins, then they begin to migrate to the opposite ends. Okay? They begin to chop away the kinetochores, like Pac-Man, eating away the kinetochores until you reach the opposite end of the cells, the anaphase. Okay? That is the whole metaphase, anaphase interface. How it goes from one to the other. Okay. Any question on this one? Yes.
Does that have anything to do with the cleavage furrow?
What was that?
The cleavage furrow, does that?
No, that is at the end. That's after the telophase. Yeah, this is for the metaphase, anaphase. The cleavage furrow, that is after this. It goes from the anaphase, then telophase, and it's cytokinesis. The cleavage furrow is cytokinesis. Okay. This one is what happens here, right here. Metaphase, anaphase. Cleavage furrow, that will be here. Telophase, cytokinesis. And those will be the microfilaments. These ones right here are the microtubules. Okay. Any question on that? Metaphase, anaphase? Okay. You guys want to do an activity for this?
Yeah.
Okay, so let's do this activity for the anaphase, metaphase, anaphase. Okay. So I need two ladies that they look similar. Look similar. Two ladies that they look similar. Who might you do? Okay, so here you have the sister chromatids. Okay, and they're going to be joined together by the cohesin. So we need one protein here, cohesin proteins. Who wants to be cohesin? Let me do a guide. Cohesin proteins. Cohesin? Yeah, okay. Are you guys fine with just like crossing arms or just like touching?
It don't matter.
Yeah. Yeah. Well, you can just like, yeah, in the middle, like touch elbows right there or close elbows, whatever, right? So here you have cohesin, sister chromatid, sister chromatid. They're identical, okay? They're identical. Now we need another proteins. Those are the kinetochores, right here and right here.
The what?
The kinetochores.
We can show points.
Hmm?
They're asking if they got extra points.
These ones.
We don't know where you're pointing at.
Right here, this one, the red
The
one.
red?
Yeah.
Oh,
Those are the
okay.
kinetochores, the proteins, okay?
So what they leave behind them?
Hmm?
They leave like.
Right
I
here.
thought that was like what squishes us, no?
They are bound to the centromere,
So
okay?
they'll be in the middle.
It should be right here. These are the, let's say, they're attached to the waist, okay? They're attached to the waist, right here. So two proteins, two guys. You, come on, and you, yes, come on, there you go. We have two kinetochores. They're attached to the waist, but do not touch the waist, okay? Just stand to the side, right there. So these guys are the proteins, the kinetochores, okay? They're bound to the waist, to the center of it. Kinetochores, proteins, okay? It's a complex, okay? Complex proteins, like that. So we have in the middle, what do we have?
Protein.
What's the name of the protein? Cohesin. Cohesin. Huh? So, cohesin, what is the role of cohesin? Joins what? Sister chromatids, right? They are joined together because of cohesin, like that, okay? So he's joining them together.
Keep it.
Now we have the kinetochores, protein complex, kinetochore 1, kinetochore 2 over here, and they're attached to the waist. Would you want to touch here the elbows? And right there. They're attached to the waist, right? These kinetochores to the sister chromatid, this kinetochore to the sister chromatid, okay? So here you have the chromosome, okay? One chromosome, two sister chromatids. And on this chromosome, we have the kinetochore right there on both ends, okay? Now we need the multiple organizing center, the centrosome, right here. Centrosome, right here. One. Okay, let's see somebody from here. You. That is a pair of centros. That is one centrosome with a pair of centros. Okay, that is one single one. So you want these two. Yeah. Centro, one pair of centros, that is the centrosome. We need another one over there on the opposite end.
Why don't you make this two?
Okay, them two. All right?
That's the one that looks like the little spider, right? Or-
Yes, that's the one that sends the microtubules. Yes. Let's perform a magnificent play. So hopefully we can win the Oscar award. We do not be slapping anybody. They make two jokes, philosophic jokes.
What are we?
You are the microtubular organizing center, the spindle core, microtubular organizing center, the centrosomes, the perocentrioles, okay, centrosome, perocentrioles. Let me show you here, okay? Right here, this centrosome right here, perocentriole. So we're doing this slide right here. Right here, these perocentrioles, you are these perocentrioles, you are these centrosomes. Okay? These ones right there, that is over there. That is centrosome right there. Perocentriole.
Okay, that was centrosome.
Yeah, both of them. This is one pole, this is One pull. That's another pull, that's another pull, okay? This chromosome right here, that's them. Sister chromatid 1, sister chromatid 2, that's them. In the middle of the sister chromatid, you have the cohescence, which is Leroy, okay? That's the cohescence right there in the middle of the sister chromatids. These black dots, those are the kinetochores. Those are Gaspar and Carlos. So Carlos and Gaspar, they are these little black dots right here, kinetochores. Okay? Those black dots right there. You see them?
He said, what are we doing now? You're gonna fill these ladies.
You are this little black guy right there.
No,
I thought he squished us.
no.
Who?
I'm
Who's
sorry.
Him.
No, he's bound to the centromere of the chromosome. That's little black dot right there. He's not squeezing you. Okay?
He's attached to you.
Yeah, he's attached to you. And he's attached to you at the centromere, that's where it's most closely attached. Maybe that's why you're thinking that they're squeezed, right? But that is the waist of the chromosome. But he's not squeezing you. He's attached to you, right there. That is the waist of the chromosome that is centromere. 1 right here, 2 right there. Sister chromatid right there, both of them right there. Leroy cohescent, it is in the middle, it's not showing you here, but it's in the middle. That is on the next slide, okay? And then we're going to do this one, okay? Right, so from each side, from each pole, they're going to send now, this one
Scooting back.
centrosome, they're going to send the microtubules. Microtubule? Another one from over there, microtubule. Okay, microtubule. Right here, so you have perocentrioles right here. They're going to begin from here and then over
Stop
there
pulling them?
to bind to him, okay? And you pull that end right there. So imagine that there are like many, okay? They're growing. So they're like many Vivianas over here, over here, and binding to him. So this microtubule is binding to the kinetochore. The microtubule never binds to the DNA. So this is the chromatic. It binds to the protein, kinetochore. Okay, it binds to it. So this one says already it bounds, right? All right, let's put the closer together over here. Let's say not you, not yet, not yet. Okay, so that one is bound. She's bound right there, right? But not on the other side. There is a lack of tension, okay? There's lack of tension right now. She's bound, we say signal, wait, okay?
Oh, that one is like.
Yes, now
Is that
they're
here?
going to send this one, now you're going to send, she comes and goes, yeah, you better send to, no, not them, you. They're going to send you, they're going to send
Okay,
you.
they're gonna send me.
Yes, go, right? And now it's going to bind over here, and again, it touches the kinetochore. Now, kinetochore here, macrotubule there, bound, kinetochore here, macrotubule there, bound, now there is equal tension. Now your guys are going to pull it into the middle, okay? Before it was not in the middle, because that she's pushing right there, they're going to place in
Do
the
I have
middle.
to push all of them?
Not yet. Not yet. Now there is equal tension because there is the other side as well, okay? Now they're in the middle. That sends the signal. When all the chromosomes, like this, are bound by the microtubules, to the kinetochore, they're going to be placed in the middle. Once they are placed in the middle, then they're going to send a signal to, over here, to the separase, okay? Separase, it's a protein that is going to cut cohescent, little one, so it's going to slap them, cut them, okay? Just take him away, just take him away, yes. There you go, okay? Separase, cut, cohescent. So now they are not bound together. So now they're going to move, but he's going to walk you. You are going to walk along many different microtubules, displacing, all the way to reach over there. So that's a good point. It is not that she's pushing, but that he's walking, okay? He's walking along
But
this
see
chain.
that picture? This tube, pulling.
That's how it looks like, pulling, but it's not pulling, okay? He is walking until it reaches to the other end, okay? Yes.
It's drifting apart, there's no like force.
Yeah, he's been removing, like Pac-Man, think like Pac-Man.
Okay.
He's removing those microtubules until it reaches to the other end. It looks like pulling, but in fact, he's the one that is eating them, like walking, displacing them. Okay? So he's the one moving all the way over there, and you're the one doing the same thing, moving there, walking through the channel of the microtubule. Think about like these microtubules as highways, okay?
Yeah.
And that he's walking, driving through the highway. Okay? And it goes to the other end. Okay? And that's what happens right there. Separation of the DNA right there. And that's what happens right here. It separates, cuts the cohesive proteins. Okay? Any questions?
Like, who was she again? What was she again?
She was this guy right here, separates. Cut the cohesive, so remove Leroy was the cohesive, gives them, and now they're able to move. To the other cement. Okay?
I have a question.
Yes.
It might be irrelevant, but it's probably relevant, but.
Can I go back there?
Considered mechanical work or something.
The mechanical work is being done by him. Yes, he needs ATP to do that, to transport, to move. Yes.
What's he again?
That he is the kinetical proteins. Okay.
Yeah.
Right, thank you. Your half, my thanks.
You're the mechanical.
Extra credit points.
How about?
I need it.
Alright, so that's the mathematics spindle, right? Separate microtubules in order to control this movement of the DNA. And during this prophase, the assembly of the spindle of microtubules begins in the centrosome. There is a microtubule organizing center. Centrosome, microtubule organizing center, that is the third centrosome. So the centrosome replicates, so it forms two centrosomes, and they migrate on opposite ends. Okay? And then these microtubule spindle fibers are going to attach to the chromatids at this protein complex on the DNA called the kinetical. Okay? That is him. What's your name again? Carlos?
Yeah.
Carlos and Gaspar. Those are the kineticals. That's where they are bound to the DNA, and that's where the microtubules bind to them. Microtubules never bind to DNA. They bind to the what? Kinetical. That will be on the exam. If you miss that, don't blame it on me. If you choose DNA binds, if you choose microtubule binds to DNA, I don't know where you got that from.
Wait,
Not
what you
here.
said? Geneticals that bind to what?
Yeah, the microtubules,
Don't bind to DNA.
yes. The microtubules will go and bind to the kinetical. They will not bind to the DNA. They bind to the kinetical. Bind, send the signal, and then kinetical moves. It carries the DNA. Okay? Microtubule grows from the microtubule organizing center, that is the centrosome, sends out the microtubules, the kinetical microtubules. It binds to the kinetical, that's why they're called kinetical microtubules. Binds to the kinetical, and then once they're caught, separates, cohesins, follow that, it can move. Separation of DNA. Okay. And that's what we're seeing right there. These are the microtubules. You have here the chromosome, the centrosome, and all that. Okay, here you have like a beautiful picture showing that DNA right here, microtubules right there, so separating DNA. Watch this video, really amazing video. It's pretty much like show you that. So watch it at your own time so you can go back and forth. Then after this, then you go to the cytokinesis. That's where the cleavage forum happens. Okay, so there is a cleavage part, forming the cleavage forum. In plant cells, you have a cell plate. These cell plates form this cytokinesis. And in fungus, you also have like this well division, including fission, bonding, and the correlation. And cytokinesis in animals is done by the microfilaments. That's what it's going to do with the cleavage forum, the division of the cytoplasm, cytokinesis. Okay? Microfilaments, again. So remember that. Microtubules, separation of DNA, that's what we see what we saw right here. They never bind to DNA, but they bind to the kineticals, and that's how they're able to separate the DNA. And microfilaments, they're the ones involved in the cytokinesis. By making the contractile ring, cleavage forum, Okay, you're going to separate the DNA. I mean, you're going to separate the cells. Okay. So cleavage 4, contractile ring, microfilaments, separation of cytoplasm. Microtubules, separation of DNA. Okay. So here you have that the cleavage 4, contractile ring, microfilaments, creating these dotted cells right there. Cytokinesis, separation of cytoplasm. In animal cells, in plant cells, you have this, the new cell plate using these vesicles from the Golgi apparatus, it will send that, these vesicles, in order to form this new cell plate that is going to become the new cell wall. Okay? So in animal cells, microfilaments, cleavage spheral, contractile ring, cytokinesis, division of cytoplasm. Animal cells. In plant cells, you have the Golgi apparatus. The role of the Golgi apparatus send this vesicle to create this cell plate and then forming the new cell wall. So Golgi apparatus important. So here you have these basically different roles of the microtubules, how they are here, involved for the separation of DNA, and then here you have the microfilaments involved in the cytokinesis, contractile. Right, so that's it for this session. Any question before we go to the activity? Alright, so one last activity. So on this one, each team will pick each member of the team will pick a phase of mitosis. The metaphase, prometaphase, the prophase, prometaphase, metaphase, anaphase, telophase, etc. Pick one, right, between your members. And do not reveal that stage to the other team members. So draw it out, your own version, right, in a paper. And then levels of the piece of paper, use that figure that I showed you. Do not write the name down.